Dear students, welcome to BioRatna. Students, every Sunday at BioRatna is a special session because it is devoted for concept building. Even just one day before the board exams or NEET exams, if you ask the students that what is their difficult chapter, if they find any chapters difficult, then most of them will point out to the genetics chapters and the biotechnology chapters. Why it should be so? Why it should be so? After your this 2-year journey starting from class 11th, now while you are appearing for exam, at that time your whole preparation should be complete. Your concepts should be clear. But it is found that the students, they find difficulty in genetics and this biotechnology. So when you go on clearing the basics of each topic of genetics, I tell you students that the next topic, the very next topic of genetics, you can predict it. You can see through it. आप बता सकते हो कि हाँ इसका अभी ये स्टेप हो सकता है. It is really that simple. And once you master genetics, then biotechnology is of course easy. So in today's session, it is for one genetics topic, that is transcription unit and gene. Our today's topic is transcription unit and this is from the chapter molecular basis of inheritance. This is your 6.5.2 in NCERT, transcription unit and gene. So here there are only 2 paragraphs. In this only simple 2, I won't say simple, there are 2 paragraphs in which is packed with so much of information. Mainly it is discussing why is it difficult to define a gene? Why is it difficult to define a gene? Fine. How do we define a gene? We say that a gene is the unit of inheritance. We say that gene is unit of inheritance which is part of DNA. Now your NCERT says that it is very difficult to define DNA taking into consideration the DNA sequences. It is very difficult to define a gene in terms of DNA sequence. 4 points are discussed there. There are 4 points which are discussed. Let us take one by one and let us see what it means. First, it is discussed that DNA, it is not just forming mRNA. वो just एक mRNA नहीं बना रहा है. The messenger RNA, which is having the information to form the protein which is going to control the characters. This DNA forming mRNA, which is forming the proteins. This is the process of this first process. This is the process of transcription. And mRNA to proteins. This is the process of translation. So it is not just the process of formation of mRNA and then by translation formation of proteins which is going to control the expression of characters. It is also the formation of tRNA as well as rRNA. It is not just not just mRNA, but also the formation of tRNA and mRNA, tRNA and rRNA, not mRNA, tRNA and rRNA, that is ribosomal RNA. So this is one point. Let us see the next point. Can we define it in such a way? If we define cistron as the part of DNA coding for a polypeptide, now let us analyze this one. Let us see how it happens in case of prokaryotes and in case of eukaryotes. In case of eukaryote students, this part of DNA, which we call gene, now it is going to this one segment, it is going to form mRNA, one mRNA. There is one more complication which we are going to discuss just now, because before mRNA also there is one precursor of mRNA. But here the point, some other point is going on. So this one mRNA and this one mRNA is going to form one protein. It is going to form protein, that too single protein. Is it so in case of prokaryotes? In case of prokaryotes, you will see that this segment, DNA segment, it will by transcription, it will form one mRNA, but this mRNA, it will form more than one proteins. This is more than one protein. एक से ज़्यादा प्रोटीन बनाता है. But of course, all these different proteins, they have related functions. They all have, these proteins, they have related functions. Related functions means, कौन सा एग्जाम्पल लेके समझाए आपको? Like this video lecture. Suppose I am preparing the video lecture, someone is editing it and someone is posting it. But all of them are working towards the same goal. So their function is related. It is not so that the different proteins, remember this, the lac operon you will be studying, it is based on this only. So all these proteins which are formed in prokaryotes from one single mRNA, they all have related functions. This is called polycistronic. This is called polycistronic gene, and this one is called monocistronic gene. Monocistronic gene. Now come to the third point. This much you understood, students? That the different way we are trying to define a gene, we are facing with some or other problem. Now let us discuss the split gene arrangement, which further complicates the definition. Now can we define it in some different manner? So like we say that it is a part of DNA, it is part of DNA which forms mRNA, which then codes for protein, Then this definition is true for prokaryotes. This definition will hold true for prokaryotes. What about eukaryotes? Let us see what happens in eukaryotes. In case of eukaryotes, this segment of DNA, this one will form the mRNA. This one will not form mRNA, This one will form the precursor of mRNA called hnRNA. What is hnRNA? hnRNA is H for heterogeneous, N for nuclear, it is heterogeneous nuclear RNA. So this heterogeneous nuclear RNA, it is not functional. This one is not functional because this RNA, it has coding as well as non-coding parts. Suppose this part is coding, this is non-coding, coding, non-coding, again coding. So let me write it E, I, and E. What does this E stand for? This E is the coding part. This E is the coding part and this I is the non-coding part. It is the non-coding part. If you take its full form, then E stands for exon. This E, it stands for exon. And this I, it stands for intron. Exon and intron. Often students take exon as the non-coding one and intron as the coding one. क्योंकि कहीं पे वो नाम थोड़ा सा confuse कर देता है. Intron, somehow the brain takes it that this is the in thing, कि यही होगा जो coding part है. Don't take it, this in को आप convert कीजिए in for interfering. So this is the interfering part. So this interfering part, as it is there in between the coding part. अब देख रहे हो यहाँ, ये exon है, ये exon है. In between there are the non-coding part. यहाँ से अगर आप प्रोसेस स्टार्ट भी करते हो, फिर जाके वो I पे जाके इंट्रॉन पे जाके रुक जाता है. So now there has to be, this arrangement is called split gene arrangement. This arrangement, students, it is called split gene arrangement, and this hnRNA, it needs a processing. It needs processing so that it can be converted into active mRNA. So this split gene arrangement, you can see how it is complicating the definition in terms of the DNA segment. आप बोल ही नहीं सकते हो कि यहाँ जो DNA का segment है, which is coding for mRNA and mRNA is going to form the polypeptide, then this part of DNA, complete part, वो तो कोड कर ही नहीं रहा है. It is having some introns, the interfering units, जो खुद काम नहीं कर रहे, exon को भी काम नहीं करने दे रहे. So you cannot define gene in this way too. So what can be the next option? Let us go for NCERT's next option. Now students, the fourth and the last point. This is the invisible point in the magical book called NCERT. There are two paragraphs. When you complete the first paragraph, you will think that the topic is over. And then you overlook the smaller paragraph below it. Even if you read it, you think that something irrelevant is written. No. Don't take any word as irrelevant. आपको ये fourth point बच्चा वहीं पे मिल जाएगा. What is this fourth point? You will see that the inheritance of characters, it is, students, also affected by the promoter and other regulatory sequences. I am first writing, then I will explain it to you. Fine. Other regulatory sequences. Mark this choice of word here. It is not here, regulatory gene is not written. It is written regulatory sequence. Now why these are called then regulatory? Why we loosely also call them as regulatory gene? So for that, first we have to see, come to your transcription unit. ये है आपका ट्रांसक्रिप्शन यूनिट. दिस इज 5(3). देन दिस वन विल बी दिस अपोजिट डायरेक्शन. एरोज आपको बनाने ही बनाने हैं. वन इज प्रोमोटर, वन इज टर्मिनेटर. एक सेशन हमारा हुआ था ना कि जहां हमने देखा था कि हाउ कैन यू नाउ डिसाइड विच वन इज टेम्पलेट एंड विच वन इज कोडिंग स्ट्रैंड. प्लीज गो थ्रू दैट संडे सेशन ऑल्सो ना. तो यहाँ पे देखो प्रोमोटर में जो 3, प्रोमोटर के पास जो 3 वाला है, दिस वन इज द टेम्पलेट स्ट्रैंड, दिस वन इज द टेम्पलेट स्ट्रैंड. दिस प्रोमोटर, देन देर आर दिस अदर रेगुलेटरी जीन्स. प्रोमोटर है, फिर ऑपरेटर है, फिर इनहिबिटरी जीन्स है. लेट अस कॉल इट इनहिबिटरी सीक्वेंस नाउ. इनहिबिटरी सीक्वेंस है. So these all they are affecting the inheritance of characters. It is not that structural part of the gene which is coding for mRNA. But these other sequences, they also affect the inheritance of characters. That's why these sequences, these are called, this is called not regular, now we call it, sometimes it is loosely, these are also defined as regulatory genes. These are also called as regulatory genes. तो यहाँ भी जीन वर्ड आ गया. तो एक्सक्लूसिवली फिर हम इसे कैसे यूज कर सकते हैं वहाँ पे. सो यू आर अंडरस्टैंडिंग द डिफरेंट टाइप्स ऑफ डेफिनेशंस एंड व्हाट आर द डिफरेंट टाइप्स ऑफ डिफिकल्टीज which are related to the DNA sequences. Fine. So this was about this 6.5.2 transcription unit and gene. I hope that this session was helpful for you in building up this much of the concept today. Students, in our Bioratna, we have started just last week, we have started the 2024 batch. So that class 11th and 12th, these students gradually, it is a 2-year journey. It is a 2-year journey. आप उसे कहीं पर भी रिलैक्स होके 3 months में, 1 month में, 6 months में कंप्लीट नहीं कर सकते हो. So it is a 2-year journey and there all the topics in the video lectures, all the points will be covered, every concept will be built. Whatever is taught in the last week, उसका कोई टॉपिक लेके उसमें कोई अगर डिफ़िकल्ट कंसेप्ट है उसे डिस्कस किया जाएगा. So that to gradually build the confidence of the students and let them understand that clearing board as well as clearing of NEET exam in 2 years, आपकी 11th और 12th की जर्नी कल्मिनेट होगी वेरी सक्सेसफुली in 2024. Of course 2023 batch will also be benefited by this because ये तो 12th का ही है, but in those 11th के lectures भी 2023 वाले students के लिए भी बहुत beneficial रहेगा because that is also in their syllabus. So if this video is helpful to you, then do subscribe. Thank you for watching the video.